Hello, students. A very good afternoon to all of you. So today we are going to have a revision session. And in this session, we are going to talk about a new chapter that is respiration in plants. All right. So before we head on to this topic, first let me do a technical poll. Please try to answer this. So I just want to know if my audio and video is clear. Yes, perfect. Let's get on to the session. So the first, as usual, before we head on to any topic, we have to answer some questions, right? We'll take a small quiz and answer some questions. So the end product of fermentation are oxygen and ethanol, and then you have carbon dioxide and acetaldehyde, carbon dioxide and water, and then you have carbon dioxide and alcohol. Anyways, so I will give you the poll for MCQ. Okay, hold on. Yeah, here is your poll. Okay, that's quick. Anyways, we have an answer. All right. So the answer you given was D, that is carbon dioxide and alcohol. And this is the right answer because when it comes to fermentation, fermentation is a type of anaerobic reaction. And the end product is always alcohol because there is an enzyme called as alcohol dehydrogenase, which converts pyruvate into alcohol and carbon dioxide. Okay. Yes. So another question. So the overall goal of glycolysis, Krebs cycle, and the TCS and the electron transport system is the formation of ATP in one large oxidation reaction, sugars, nucleic acids, and ATP in small step units. Okay. So this is a question. I will relaunch the poll. Hold on. Yeah. So here is your poll. Yes, yes. So I'll give you 30 more seconds. Okay. So try to answer this. What is the overall goal of glycolysis, Krebs cycle, and the electron transport system? We have one answer, one more to go. Students from the YouTube, you can answer through your chat box. Okay. Always. Okay. So we have one more answer in the chat box. So yeah, let's get back to the question. The question is asking, what is the overall goal? See, people, where glycolysis is a multi-step process, isn't it? Same goes with the Krebs cycle. And then we are going to study electron transport chain. Okay. We'll try to do that because that is one of the important aspects of this chapter. So anyways, so all these things are happening in a very elaborate cycles. You can see that from cytoplasm to the mitochondria and what happens in the matrix as well as the intermembrane system. In all the cases, what is happening, there is a stepwise reduction of glucose so that the energy is conserved, okay, into ATP molecules as it moves on from one step to another step. So when there is immediate, what do you tell, extraction, for example, if it was only one step process of, gaining the energy from glycolysis, so there would be tremendous amount of heat released. So that would result in a lot of problems. That means the energy cannot be conserved. To conserve energy, they usually all this process, includes production of ATP in small stepwise units. Okay, so the D is the answer. So yes, people, good afternoon. So anyways, can we go on to the topic? A quick studies we will do today. Okay, so that we will cover maximum. So let's get back to the chapter, people. So in this chapter, as we're talking about respiration, so we know that respiration and breathing are correlated. And when it comes to animals, we know we breathe, and then there is, the gaseous exchanges takes place between the tissues. And the same goes with even plants too. The plants do not, they do exchange of gases through stomata. And as they do exchange of gases, one of the primary reasons that they require this gas in the form of carbon dioxide and oxygen is for photosynthesis. Carbon dioxide is required for photosynthesis and oxygen is required for respiration process. So what is respiration? Respiration is breakdown of food material, basically. It might be carbohydrates, it might be fats, it might be proteins, okay? So in most cases, we consider as glucose. Why? Because carbohydrates are the primary source of energy. So what do? We use glucose and then we try to, break this down or reduce these sugars into carbon dioxide, water, and energy. And the energy that is converted is in the form of ATP. So this is called as a respiration process. So when you see respiration process, as I say, it is an elaborate process when it comes to eukaryotic organisms, okay, even as well as prokaryotes, but we consider eukaryotes. Why? Because it's very easy to contemplate the same thing even with us, right? So anyways, so once we have the glucose undergoes a process of glycolysis to give pyruvate. And this pyruvate, based on aerobic or anaerobic, means if it is an aerobic respiration, the pyruvate undergoes aerobic respiration through Krebs cycle and electron transport chain to result in ATPs. Whereas when you talk about the pyruvate entering into the anaerobic cycle, right? So anaerobic cycle happens without oxygen, right? So when it happens without oxygen, we call it as fermentation reaction. And we have two types of fermentation reaction, that is lactic acid fermentation and alcohol fermentation. So however it is, the primary reasons why we do cellular respiration is because to produce energies, right? To break down the glucose and produce energy. So let us see what is happening in cellular respiration, starting with our primary ingredient, that is the glucose. Okay. So once we have the glucose is stored in, if you talk about the plants have the glucose stored in the form of sucrose, which is a disaccharide. So the sucrose will be broken down into glucose and fructose initially. Okay. So this reaction, the breaking down of sucrose into glucose and fructose, again, it is catalyzed by enzymes. Okay. That is called as exokinase. So before we head on to this process of glycolysis, what is glycolysis basically? Glycolysis do not require oxygen. That is the first thing you have to remember. So it does not require oxygen. So who does the process of this reduction of this glucose molecule? So when there is no oxygen, actually it undergoes a very gradual steps of enzymatic oxidation reactions. So the enzymes do the oxidation reaction instead of the oxygen itself. Okay. So this series of enzymatic reactions is called as glycolysis. And this glycolysis is popularly known as EMP pathway. So why do we call it as EMP pathways? Because there were like three scientists called as Emden, Meyerhoff, okay, and Parnas. These three scientists, they try to elucidate the entire pathway of glycolysis, okay, and that is why it is also called as EMP pathway. Okay, so as I said, the glucose is extracted from the sucrose by breaking down this disaccharide into glucose and fructose. After we have, okay, let me go here directly. Okay, after we have the glucose will be, okay, converted into glucose 6-phosphate by an enzyme called as hexokinase. Okay, so this hexokinase, you will utilize one ATP molecule. So the ATP will be broken down into ADP plus phosphate. Okay, so this phosphate molecules will be added to your glucose at the 6th carbon atom. So when you add, see, glucose is having 6 carbon atoms, people, as we know already, correct? So at the 6th carbon atom, we add a phosphate. So that will become what? Glucose 6-phosphate. All right. So remember this, the conversion of glucose to glucose 6-phosphate is an irreversible reaction. Once this hexokinase does this job with the utilization of 1 ATP, so it's done. You cannot go back from glucose 6-phosphate to glucose. It's not possible. Now what happens? There are like series of reversible reactions. Now the glucose 6-phosphate is converted into fructose 6-phosphate. And here the glucose and fructose are isomers, right? They have the same number of number of atoms, right? But their structural formula, that is structure is different. All right. So when we talk about glucose 6-phosphate to fructose 6-phosphate, it is again catalyzed by an enzyme called as phosphoglucose isomerase. We just call it as isomerase. Okay. So the isomerase will convert glucose 6-phosphate into fructose 6-phosphate. Now, once we have fructose 6-phosphate, again, one more molecule of ATP is invested. The same reaction happens where the ATP will be broken down into ADP and phosphate. Now, here, the first case, the phosphate was added in the 6th carbon atom. Here, along with the 6th carbon atom, we add one more phosphate at the 1st carbon atom. Okay. So that will give a rise to a molecule called as fructose 1,6-bisphosphate. Okay. Now, once we have fructose 1,6-bisphosphate, now the fructose will be broken down into three carbon containing compounds, and this will be catalyzed by another enzyme. Okay, so another enzyme, this is called as aldolase. When fructose 1,6-bisphosphate is broken down into 3 carbon atom groups, so that will give rise to glyceraldehyde 3-phosphate. Okay, glyceraldehyde has 3 carbon atoms again. Okay, and it has the phosphate at the 3rd position. So that is why we call it as glyceraldehyde 3-phosphate. See, either it can become dihydroxyacetone phosphate, or it can also become glyceraldehyde 3-phosphate. Okay, to see, it is reversible reaction, right? So either way, this conversion of glyceraldehyde 3-phosphate into dihydroxyacetone phosphate or dihydroxyacetone phosphate into glyceraldehyde 3-phosphate is again, it can be isomerized by an enzyme called as triose phosphate isomerase. Okay, so only rearrangement of atoms will take place whenever there is an enzyme called as isomerase. All right. So now, since we have 6 carbon atom, when we break down 6 atoms into 2 groups, we will have 3 and 3. That means we'll get 2 molecules of 3 carbon atom containing compounds, right? So that is why we'll get glyceraldehyde 3-phosphate as 2 compounds. Okay, two in number we will get. Now, what will happen? This glyceraldehyde 3-phosphate, look at this. When the glyceraldehyde 3-phosphate is present, then an NAD plus is reduced to NADH. Okay, so this reduction reaction takes place in the presence of glyceraldehyde 3-phosphate dehydrogenase. Okay, so when a hydrogen atom is added to NAD plus to NADH, okay, now the glyceraldehyde 3-phosphate will be converted into 1,3-biphosphoglycerate. Okay, so one more phosphate group gets added to this glyceraldehyde at the 1st position. Got it? Now this 1,3-biphosphoglycerate, now till here, see people, till here we have been investing energy. Now is our return stage, correct, right? Investment and return stage. Now, once we have this 1,3-biphosphoglycerate, correct? So this 1,3-biphosphoglycerate, now what happens here? By the 7th enzyme, that is phosphoglycerate kinase. Whenever the kinase comes, now what happens? One phosphate molecule is really, removed out of this particular structure. So this free inorganic phosphate will be added to your ATP. Okay, when it is added, it will be added to your ADP. When it is added to your ADP, it will become ADP plus phosphate will give what? ATP. So here we are getting energy. Are you getting my point? Now, once we remove one phosphate from 1,3-biphosphoglycerate, we will get 3-phosphoglycerate. Okay, so this 3-phosphoglycerate will be again converted into 2-phosphoglycerate by an enzyme called as phosphoglycerol mutase. Okay, so 3-phosphoglycerate will be converted into 2-phosphoglycerate. Now the 2-phosphoglycerate, hold on, when the 2-phosphoglycerate will be dehydrated by an enzyme called as enolase, it will become phosphoenol pyruvate. Again, phosphoenol pyruvate with the action of pyruvate kinase. Now what will happen? The remaining phosphate molecule will be removed by phospho, that is phosphate group will be removed. Now all you're left with is pyruvate. Okay. So once we remove this phosphate group, again the ADP will be converted to ATP. Got it, people? So finally, what is the net product we are getting? We are getting pyruvate. First thing what we are getting is pyruvate. How many pyruvate we are getting? 2 molecules of pyruvate we are getting. And what is other things we are getting? We are getting ATPs. Here we are getting 2 ATPs. Here we are getting 2 ATPs. Understood? And what is another thing that we are getting? We are getting 2 NADH plus 2 H plus we are getting. Understood? What are we investing? 2 molecules of ATP. So glycolysis can be summarized as you have glucose, right? Breaking down of glucose will require 2 ATP is invested, correct? No? We'll invest 2 ATP. And what do we give? What do we gain? 4 ATP is gained. And then we gain 2 NADH, okay, plus 2H plus, correct? And then what do we gain? Correct? No? And then we'll gain 2 pyruvate. Correct? No? Pyruvate. Sorry. Yeah. Okay, pyruvate. So once we get this, the glycolysis is complete. Okay, here only it is. Why did I write? So 2 ATP, 2 pyruvate, and 2 NADH is obtained from glycolysis process. Now I told you that we gained 4 ATP. And why did we write 2 ATP here? Because in the initial stage of glycolysis, we would have invested 2 molecules of ATP. No? So always investment we have to count. So 4 minus 2 is 2. So this will end glycolysis, okay? See, the glycolysis reaction is where it is happening. It is happening in your cytoplasm, okay? So once you see the glycolysis process, which is happening in the cytoplasm, this is only halfway, all right? Only halfway we have reached the cycle. Again, there is a lot of things that has to happen to gain, ATP molecules. See, when we, first we'll talk about aerobic reactions because this is very, very important. And then we'll talk about anaerobic reactions, okay? In most of the cases, in all living organisms, in complex beings, we use aerobic reaction only. Why? Because aerobic reaction will give rise to a tremendous amount of ATP yield compared to that of any other process that is happening with respect to respiration. Understood? So if you want to break down the glucose completely, okay, if you see that I want to break down completely whatever the energy molecule, your food substances to gain energy, it has to undergo not just one step. Glycolysis will not accomplish the entire process, okay? There are series of other steps also that has to be accomplished so that there is complete breakdown of food material. Let us see one by one. We understood that the glucose that is produced, that is formed or that is present in the cells undergoes glycolysis or EMP pathway in the cytoplasm. So first step of the breaking down of your food is done. What is the second step? See, once we accomplish the glycolysis, what we get is pyruvate. Understood? So this pyruvate from the cytoplasm is transferred all the way to the mitochondria. So in mitochondria, we have 2 regions, people. So you know that the mitochondria is a double membrane system. So in mitochondria, we have outer membrane and inner membrane, correct? So between this outer membrane and inner membrane, there is a space called as intermitochondrial space, correct? So the inner membrane, if you see, it has numerous folds, which called as crista. So which divides the mitochondrial matrix into, the folds like this, the inner membrane folds. And then you have the medial portion called as matrix, okay? So this middle portion of the mitochondria is called as matrix, isn't it? So in matrix, what happens? There are specialized enzymes, or it undergoes very sequential enzymatic reactions. Okay. So here what happens in the initial stage, as your pyruvate from the cytoplasm is entering into your mitochondrial matrix, okay, the pyruvate undergoes a step called as oxidative decarboxylation reaction. Understood? It undergoes oxidative decarboxylation reaction where your pyruvate, see pyruvic acid, okay, in the presence of coenzyme A, there are several coenzymes, and CoA and NAD plus in the presence of some of the cofactors like your magnesium ions and an enzyme called as pyruvate dehydrogenase. So this pyruvic acid is converted to acetyl CoA. Understood? So first what happens? Pyruvic acid from the glycolysis is utilized, and it is moved to the mitochondrial matrix. In mitochondrial matrix, what happens? So the enzymes like CoA, NAD plus, okay, and then again we have pyruvate dehydrogenase and Mg plus is utilized to convert your pyruvic acid into acetyl CoA. Understood? Next, you have your NAD plus is again reduced to NADH plus H plus. Understood? So we have one more NADH here. You have to remember this. Understood? So this reaction is called as oxidative decarboxylation reaction, and this is also called as a gateway reaction. Why? Because from here only it undergoes another important cycle called as TCA cycle or Krebs cycle. Clear, people? So you remember this step. This is a very important step where we always forget. Okay, pyruvic acid is converted to acetyl-CoA in mitochondrial matrix. Very simple. So this conversion of pyruvic acid into acetyl-CoA in your mitochondrial matrix is accomplished by who? It is supported by an enzyme called as pyruvate dehydrogenase. And also there are coenzyme A, several coenzymes are required, as well as your NAD plus is reduced to NADH plus H plus. Understood? So this step is complete. Next one is the products of your oxidative decarboxylation is your acetyl-CoA and NADH, correct? No. So this acetyl-CoA and NADH enters this cycle called as Krebs cycle or TCA cycle. Understood? So once it enters the Krebs cycle or TCA cycle, now here what happens? The hydrogen atoms that is present in your remaining chains of your glucose, which is already broken down into pyruvate, right? So the three carbon atoms, whichever these hydrogens are attached, a sequential removal of hydrogen atoms takes place in the Krebs cycle. We'll talk about this. So these hydrogens are utilized for the process called as oxidative phosphorylation, where the bulk production of ATP happens. Understood? So we have completed 2 steps here. Let us see what is the third step, that is your Krebs cycle. Okay, let us go for Krebs cycle. Fine. Look here, people. Pyruvate is converted to acetyl-CoA. You can see how the structure is changed. Correct? No? One carbon dioxide is removed from the pyruvate. Correct? No? So this removal of carbon dioxide happens, and when the carbon dioxide is removed, okay, now what happens? Your pyruvate is converted to acetyl-CoA. Understood? And this enzyme, pyruvate dehydrogenase, is very important. And I told you there are several coenzymes required. For example, CoA-SH, and you have, TDP, TPP, and FAD. It's not necessary, but just remember that it contains a lot of enzymatic complex. Understood? Fine. Let us go for your citric acid pathway. Okay. So is it clear, people? Until here, are you understanding? Do you have any problem? Give me a quick yes or no. Faster. Did you understand till now? Okay. Let's get back to 1 second. Yes, now we can start here. Okay, if you can just use the chat box and you can tell me if you didn't understand. Okay, fine. Before we head on to this, let me just zoom it. Okay, it's too big, isn't it? Yeah, it's all right. Little bit I will reduce. Okay, sorry. 1 second, people. Come on. Okay, I hope you can see the screen. Okay, at least this much. Okay, yeah, let it be like this, and here, yeah, proper. Okay, yeah. So let us get back here and understand. So what is happening here? The initial molecule, or the initial. Oh, we can't use the pen. Okay, sorry, people. I'll just go back again. Yeah. Anyways, yeah, the initial molecule that we had from the the second reaction. What is the second reaction? The second reaction is called as oxidative decarboxylation of pyruvate. Correct, right? So when oxidative decarboxylation of pyruvate happened in the mitochondrial matrix, what did we get? We got acetyl-CoA. Correct? No. So this acetyl-CoA will enter a cycle called as TCA cycle. Why we call it as TCA cycle? It is called as tricarboxylic acid cycle. Okay. So tri means 3 carbon atom containing carboxylic acid. Basically, they will have COOH group. Anyways, so this tricarboxylic acid is also popularly known as Krebs cycle, because Hans Krebs is a scientist who discovered it. All right. So anyways, so now when we have your we have your acetyl-CoA. So when acetyl-CoA have to enter
the tricarboxylic acid cycle, first it starts with your oxaloacetate. Okay. Initially, to even start the process, now what happened? The condensation of oxaloacetate will take place. When the oxaloacetate is condensed, okay, this will convert this acetyl CoA into a molecule of citrate in the presence of citrate synthesis. Okay. Citrate synthase is an enzyme that converts your acetyl CoA into citrate, okay, by condensation of oxaloacetate. Okay, you understood? Once you have citrate, the citrate will undergo a series of isomerization reaction. Understood? So in this series of isomerization reaction, first of all, the citrate has to be converted to isocitrate. This is the ultimate goal. So directly, it is not possible to convert citrate into isocitrate. That is why what happens, there is an intermediate enzymatic reaction. First, initially what happens, the citrate will be dehydrated. One water molecule will be removed by an enzyme called as aconitase. Okay. Once you remove one water molecule out of the citrate, okay, this will become cis-aconitate. Understood? Again, for the cis-aconitate, when the water molecule is added, now the rearrangement of the structure will take place. Okay. So this aconitase is an enzyme. So cis-aconitate will be converted to isocitrate. Okay. What happened? Oxaloacetate got condensed. When condensation happens, acetyl CoA will be converted to citrate. Once we have citrate will be dehydrated to produce aconitase. Correct? No. Again, when you have citrate will be converted to cis-aconitate with the help of aconitase enzyme, and here water molecule is removed. Now your cis-aconitate will be converted to isocitrate by addition of water. Okay. Now, once we have isocitrate, what you have to remember here is, again, the NADP plus is reduced to NADH plus H plus. So once you reduce your NADP, now hydrogen atoms from the isocitrate is removed. Okay, we get this hydrogen atom. This is very important. Understood? So once you remove the hydrogen atoms from the isocitrate, your isocitrate dehydrogenase is an enzyme that is actually helping in this process. When we remove hydrogen atoms out of your chain, okay, 3-carbon chain, now a carbon dioxide is released. Okay. Now your isocitrate will become alpha ketoglutarate. Now alpha ketoglutarate is again, okay, reduced by, see again what happens, NADP plus is reduced to NADH plus H plus. So this is the second place where the removal of hydrogen will take place. Again, with the removal of hydrogen, one more carbon atom is taken out of the structure. Clear? Okay. So now this reaction is catalyzed by which enzyme? Very important enzyme, alpha ketoglutarate dehydrogenase. Wherever dehydrogenate comes, you can see that there is a removal of hydrogen and carbon dioxide. Understood? Okay. So once we have alpha ketoglutarate being reduced, now what we'll get? We'll get succinyl CoA. Understood? In the process of converting this succinyl CoA to succinate, okay, now what happens? A GDP, okay, gynosin diphosphate is converted to GTP, that is gynosin triphosphate. This is a very spontaneous reaction. Okay, what is happening? Once we have GTP, the GTP is again broken down into GDP plus phosphate, and again this phosphate is used for ADP plus phosphate to become ATP. Understood? So we'll get an ATP here. Got it? So it's a coupled reaction, we can tell. I will tell you. Once we have succinate, again, see here, succinate for the conversion of oxaloacetate. Okay. Initially, what happens? There is an enzyme called as succinate dehydrogenase, where your FAD will be reduced to FADH2. Okay, when the two hydrogen atoms are removed from this structure, the succinate will become fumarate. Okay, with the addition of water molecule in the presence of fumarase enzyme, fumarate will become malate. Understood? Again, removal of one more hydrogen atom from your malate or reduction of NAD plus to NADH. What does it gives? It gives rise to oxaloacetate. Understood, people? So ultimate goal is alpha ketoglutarate, when it was reduced to succinyl CoA, now the succinyl CoA will aim to become oxaloacetate. Why oxaloacetate is necessary? Oxaloacetate is the preliminary requirement, okay, or very important requirement for the beginning of citric acid cycle. Understood? Or TCA cycle. Did you understand, people? Can we do a quick revision? So what is happening here? First, we had acetyl-CoA. Acetyl-CoA was converted to, citrate by the condensation of oxaloacetate. So that will give rise to what? It will give rise to cis-aconitate. Or you can just tell that once we have citrate, it will be converted to isocitrate. Understood? So once we have isocitrate will be reduced to alpha ketoglutarate by converting NAD plus, by reducing NAD plus to NADH. Understood? So once we have this, we have one release of one carbon dioxide molecule and one molecule of NADH. Clear? Again, alpha ketoglutarate will be converted to succinyl-CoA. Again, one more time, NAD plus is reduced to NADH plus H plus. Understood? Again, here elimination of one more carbon dioxide will take place. Here what happens, the succinyl-CoA will try to become oxaloacetate. Okay. So here during this process, succinyl-CoA is converted to succinate or succinic acid. So here the succinate conversion of succinyl-CoA and succinate will result in production of one GDP molecule. So this gynosin triphosphate, okay, is coupled with production of ATP. The produced GTP will be broken down into GDP plus phosphate, and this phosphate will be used for the production of one ATP molecule. Understood? So this succinate will be again reduced to fumarate, okay, by reduction of FAD to FAD plus, and FAD to FADH. Okay. Next, fumarate will be converted to malate by addition of one water molecule, and then malate will be converted to oxaloacetic acid. Again, in this process, an NADP plus is oxidized. Okay. NADP plus is reduced to NADH and H plus. Did you understand? So what can we conclude? Each pyruvic acid molecule produces how many NADH? 4 NADH and H plus, and one molecule of FADH2 and one molecule of ATP. Did you understand how do we get this ATP? This one. Clear? Fine. So you understood that by the end of this reaction, okay, that is for every glucose molecule that is broken down, okay, to release carbon dioxide, okay, it will produce 8 molecules of NADH plus H plus and 2 molecules of FADH2 and 2 molecules of ATP. Did you understand? How did we get 2 molecules of ATP? Here I told it is 4, correct, right? How did we get 8 here? You should understand here the acetyl-CoA is 1. We are talking about one acetyl-CoA. The pyruvate is produced by the glycolysis is 2. When pyruvate is 2, acetyl-CoA is 2. So you double this cycle. Okay, you double it. So when you double it, ultimately for every glucose molecule, we will have how many? We will have 8 NADH and H plus ions, and then we will have 2 molecules of FADH2 and 2 molecules of ATP. Is it clear, people? So this NADH and FADH2 are very, very, very important. Okay. Why? Because these which carry the hydrogen ions with them are very responsible for oxidative phosphorylation for the production of ATP, okay, in your ETC, in electron transport chain reaction. Clear? Okay, fine. Now, before we head on to electron transport chain or ETC, here is your first question. Which statement is wrong for Krebs cycle? There is one point in the cycle where FAD plus is reduced to FADH2. During the conversion of succinyl-CoA to succinic acid, one molecule of GTP is synthesized. Okay, the cycle starts with condensation of acetyl group, that is acetyl-CoA with pyruvic acid, okay, to yield citric acid. Okay, next one. There are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. Now, people, I will launch poll, okay? You can answer this question. What is happening? Yeah, fine. You have 1 minute, try to answer this. And more quicker is more better for us. Okay. Which of the following is wrong statement? Okay. Read the question properly. There is one point in the cycle where FAD plus is reduced to FADH2. Okay. Come on, others. Give your answer. Let me check on the YouTube people. Yes, you have 10 seconds. Your time is up. Okay. Fine. I will end the poll. One have answered A, some have answered D. Okay. So let's see if you are right. Okay. See, people, the first statement says there is one point in the cycle where, okay, where your FAD plus is reduced to FADH2. Is it right? Can we go back? Okay. How many places we can see FAD being utilized? Okay. We can see that it is one place, correct? No, here FAD is used for FADH2. Okay. Is there any place that we can see FA? It's not possible. No, you can see here. Is it there? No. So that means whoever answered A, can you see the second place where you have used FAD? One, correct? No, it's only one place. Is there any other place? Definitely no. That means your statement one is correct, isn't it? Fine. Second option. During conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. Let's get back. See, succinyl-CoA to succinate or succinic acid, what is synthesized? GTP is synthesized. I told you it is coupled with ATP synthesis also. Correct? No? That is correct. Fine. Third option. The cycle starts with condensation of acetyl-CoA with pyruvic acid to yield citric acid. Yes or no? First, what happens? Condensation reaction will take place, isn't it? Condensation of pyruvate will take place. See, it is condensed with pyruvic acid. They have asked, correct? No. So you will let us keep this point like that. So let us see the final option. There are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. Is it right? So are we using here NAD plus 1? Are we using here NAD plus 2? And are we using here NAD plus? Correct? No, 3 points. Fine. What is the third option? Condensation of acetyl group acetic CoA is correct. But is it condensed with pyruvic acid or is it condensed with your oxaloacetate? That is why oxaloacetate is very essential, isn't it? So your third option is wrong. Okay, your fourth option is correct. Got it, people? So oxaloacetate is used for the condensation. You can see here, no water molecule is added for this. Okay, so that will condense it into citrate. Where is the pyruvate here? Pyruvate is not participating here because pyruvate is already converted to acetyl-CoA. Understood? Clear? Fine. So let us go back to another question. I'll launch the poll in the beginning. Okay. During which stage in the complete oxidation of glucose are the greatest number of ATP molecules formed from ADP? Glycolysis, Krebs cycle, conversion of pyruvic acid to acetyl-CoA, or electron transport chain? Where is the highest number of ATP yield happening in the complete breakdown of glucose? Glycolysis. What about others? Give your answer. Come on, people, make it fast. Okay. So 1 minute up already. Fine. I will try to end the poll, people. It's because already 1 minute end poll. Okay. See, the glycolysis will yield how many ATP molecules, people? Okay. We had only 2 ATP, right? Correct, no? In Krebs cycle, how many ATP we are getting? 1. Are we getting any ATP from the conversion of pyruvic acid to acetyl-CoA? Not at all. Whereas when it comes to electron transport chain, okay, almost 36 ATP molecules are coming from eukaryotes and 38 ATP molecules are coming in prokaryotes. I'll write it here, prokaryon, and this is eukaryon. Okay. You can see the amount of yield. 90% of the ATP yield that is coming by the oxidation of your glucose comes from electron transport chain. Now you can blame me. Why? Because electron transport chain is not being taught yet. Correct? No? So here it is. Let's see what is happening in your electron transport chain and how this chain helps in production of tremendous amount of energy. Okay. What is oxidative phosphorylation, people? See, oxidative phosphorylation, the main goal that our pyruvate entered into our Krebs cycle was to make sure it produces enough amount of hydrogen. And this hydrogen is used for the transport of electrons, okay? And it is coupled with oxygen. That is why we get water molecules out of glycol, like breakdown of glucose, correct? No. When we write glucose breakdown formula, we'll write what? C6H12O6 plus O2, okay? Will give rise to what? Will give rise to carbon dioxide plus water plus ATP variety. We know where the carbon dioxide comes from. But do we know where do we get the water from? We are getting the water from the hydrogen and oxygen. Why do we use oxygen? We use oxygen so that we can couple these hydrogen atoms which are used for the transport of electrons with oxygen, okay, so that the chain of reaction is complete. Okay, so what is happening here in electron transport chain? So where does it take place in the first case? So the electron transport chain primarily takes place in the inner membrane of crista of mitochondria. You have two membranes, outer membrane and inner membrane. So in the inner membrane of crista of mitochondria, there is a chain reaction of transport of electrons, which ultimately results in the production of ATP. So this is called as electron transport chain. Understood? So what is happening? Let us see. So in the beginning of the electron transport chain, first of all, your NAD plus is oxidized to, see, NADH plus H is oxidized to NAD plus. So when it is oxidized at the complex 1, what is your complex 1? Complex 1 is containing an enzyme called as NADH dehydrogenase. Okay. So we have several complexes that are present in the membrane of your mitochondria. Which membrane? Inner membrane of your mitochondria. They are having specialized enzymes to take, to undergo a specific process. Understood? Now, what is happening when an NADH is oxidized to NAD plus? Okay, so that is the removal of hydrogen will take place, isn't it? So that will result in the production of two electrons. Understood? So these two electrons with a complex, other complex like FMN complex and FES, okay? So they will help in transportation of electrons from complex 1. Okay, they will move these electrons, okay, through the complex 1 and it will reach here. Okay, let us wait here. It has reached. Okay, from the complex 1, electrons are liberated. Let us say like that. Understood? After the electrons are released from the complex 1, you can see here there is one more complex. Okay, this complex is called as, okay, see here what is happening in your complex 3, correct? No, in your complex 3, first let us go to complex 3 and then we'll come back to what is happening in complex 2. See here in complex 3, what is happening? You have few more. Okay, there are electron transport system called as cytochrome B6. Okay, this cytochrome B6, okay, will again produce some of the electrons. So actually this particular reaction of transport of electrons is coupled reaction. Okay, so what is happening initially? See, this ubiquinone is there. No, UQ is for ubiquinone. Ubiquinone is converted to ubiquinone hydrogenase. Understood? So this ubiquinone hydrogenase is transported to cytochrome, complex 3 or cytochrome B6. Again, when it is converted, you can see here the electrons are coming from conversion of succinate into fumarate. Okay, and again the electrons are coming from conversion of NADH to NAD plus. So these electrons which are transferred from complex 1, okay, and complex 2, you can see here complex 2, okay, these both electrons are carried by ubiquinone. Okay, ubiquinone will carry these electrons. As it carries the electron, ubiquinone is converted to ubiquinone hydrogenase. Understood? So that will enter complex 3. Got it? So this complex 3, when we talk about, it is a very small protein complex which is present in the inner membrane of the mitochondria, and it is a mobile complex. What is this mobile complex? Mobile complex means it can transport the electrons from one complex to another complex directly. Between what it is transferring, it can transfer electrons from complex 3 to complex 4 and complex 5. Did you understand, people? So this mobile movement of electrons is accomplished by who? Complex 3. Now what is happening? The electrons that is gained by the complex 3, okay, is again transferred through cytochrome C. Understand? So the cytochrome C, what it will do? It is a mobile unit that carries the electrons from your complex 3 all the way to your complex 4. Now what is there in your complex 4? Complex 4 has again electron transport units called as cytochrome, okay, co-oxidase, cytochrome, this one is very important, cytochrome A1, cytochrome A3, okay, and they also process 2 copper centers, understood? So they accept these electrons, okay, when they accept these electrons, okay, they will remove this hydrogen, correct? Now hydrogen is coupled for the movement of electrons, no? So they will remove these few molecules of hydrogen by adding this hydrogen to oxygen. Understood? So this is done by cytochrome C oxidase enzyme. So once this is done, now all these hydrogen ions that are channelized, okay, now what will happen? They start moving into your final complex called as complex 5. So if you remember, one of the concepts that we discussed, that is called as, you remember, we had a hypothesis when it comes to thylakoids, correct? No? So in this hypothesis, we talked about how the electron transport system takes place for the formation of, ATP molecules, correct? So in this hypothesis, we discussed 2 important factors called as F0 and F1. You remember? Okay. So when we are talking about thylakoid, correct? No, in the membrane of thylakoid, we had something called as ATP synthetase. Correct? No. So ATP synthetase has 2 units. What is the 2 units? To the inner, you see, let us take that this is inner, this is outer, towards the matrix. Okay. To the inner side of your mitochondria, what is there? There is F0 complex. To the outer side, what is there? There is F1 complex. Now, what happens as the hydrogen ions moves through this ATP synthetase, that will lead to conformational changes. So these conformational changes will make your F1 complex, okay, or a protein complex that is present in your outermost surface of your inner membrane. Now it will convert what? It will start adding ADP to a free, inorganic phosphate to produce ATP molecules. Understood? So for every ATP that has to be produced, at least 4 hydrogen atoms have to pass through your ATP synthetase. So by this, the continuous, production of ATP will take place. Can we summarize once again what is electron transport chain very quickly? Okay. What does electron transport chain involves? Electron transport chain involves a complex of electron carriers, okay, series of electron carriers. So what are those electron carriers? First of all, this electron transport system is present where? It is present in the inner mitochondrial membrane. So here the byproducts from your prior reaction. What is your prior reaction? Krebs cycle. What did we get from Krebs cycle? We got the important factor, that is NADH and FADH, correct? No? FADH2 and NADH2. So this FADH2 and NADH are very, very, very important. Why? Because they act as carriers of electrons. Understood? Now, initially what happens? The FADH that is produced in your Krebs cycle is used in complex 1 by an enzyme called as NADH dehydrogenase. So what will happen? NADH is oxidized to NAD+, that it will result in free hydrogen atoms, okay, and also electrons. So these hydrogen atom, okay, and electrons are used for the movement of electrons. You can see as the electrons are moved across, now what will happen? As the hydrogen is moved across, that will result in movement of electrons from one complex system to another complex. Okay. Now when we look at the other complex, what is our other complex? Our third complex. Understood? Our third complex, okay, they see, when you look at the third complex, look at this, it is coupled with the first complex and the second complex. How it is coupled here, okay, when the FADH is reduced to FAD, okay, in this process, what is happening? The electrons that are released here is again, okay, transferred by reducing this ubiquinone to ubiquinone hydrogenase. Understood? Again here, ubiquinone is reduced to UQH, correct? No. So here what happens? Both the cases UQ is converted to UQH. That will result in transfer of electrons from your complex 2 and complex 1, and it reaches complex 3. What does your complex 3 contains? Complex 3 contains again, okay, it is a small protein unit which is present in the inner membrane of mitochondria, and this has a capacity for the transfer of electrons. Understood? And this transfer of electrons happens through who? It happens through some of the complexes called as cytochrome BC complex, and it also contains cytochrome B and cytochrome C. Now this complex 3 is called as a mobile complex, okay, or a mobile electron transport complex. Why? It takes the help of cytochrome C and it transfers the electrons from the complex 3 all the way till the complex 4 and 5. Did you understand? So during this process, what happens? When the electron reaches from complex 3 to complex 4 has again electron transport system, something like, A1, A3, and they also have copper centers. Understood? Here what they will do, they will use this hydrogen atom, okay, and they'll couple it with your oxygen atom to produce water molecule, okay, and this is done by an enzyme called as cytochrome oxidase. So the remaining hydrogen atoms, okay, that are moved across, see, from here to here, from here to here, from here to here, and all the way they reach where? They reach your complex 5. As the hydrogen atoms are reaching your complex 5, your complex 5 is made up of what? It is made up of two important regions. One is called as F0 region, other one is called as F1 region. Now, as the hydrogen atoms move through the F0 region, it leads to conformational changes in the ATP synthetase. Understood? So these conformational changes will result in, okay, addition of free inorganic phosphate to your ADP to produce what? ATP molecules. Understood, people? So this is called as oxidative phosphorylation. So these are the important steps that are happening in oxidative phosphorylation. What is that? One is your NADH plus H plus, okay, where a redox reaction, where oxidation reaction will take place, that will result in production of electrons. Okay, from complex 1, there what is produced? 4 hydrogen atoms are produced. Okay, they're pumped through the matrix. As they're moved through the matrix, the electrons are transferred. See, next one, FADH2 is reduced at the complex 2, correct? No. So here again, the electrons are picked up by, the same complex that transfer both complex 1 electrons as well as complex 2 electrons, that we call it as ubiquinone. Understood? So from the complex will carry 4 protons into the matrix, into the intermembranous space. That will carry what? It will carry the electrons from complex 3 to complex 4. I told you this is a mobile unit. From complex 4, again the 2 protons are pumped into the matrix, where you have 20% of the hydrogen that is pumped will become water, and rest of it is used for the movement to the complex 5. Understood? So once we move the protons through the complex 5, now these protons will change the conformation in the ATP synthetase, that will result in formation of ATP by addition of phosphate to your ADP. So the energy is produced. You got my point? So if you see, the overall reaction of the glycolysis will result in, okay, your glycolysis, all that will produce only 4 ATP molecules, correct? No. Whereas 38 ATP molecules are produced by your glycose, okay, through the complete oxidation of, through the complete oxidation of your glucose content, okay? So overall result is 38, correct? No. In that, 4 of it is coming from who? 2 is coming from glycolysis, 2 is coming from Krebs cycle, correct? So remaining 34 is produced by who? This one, electron transport chain or oxidative phosphorylation. Understood? So 90% of ATP comes from who? Electron transport chain. So the question that was asked is answered here, correct? No, people? And who are the electron carriers? NADH and FADH2. You have to remember this. Understood? Fine. So the question is let me finish here. Okay, let me we have No time. Sorry, people. Okay, I will conclude with this slide. Okay, and the rest of it is very easy. Fermentation reactions. That's all. So when we look at this overall reaction, what we can conclude from this is the glycolysis will result in the overall glycolysis will result in 8 ATPs. Oxidative decarboxylation will result in 6 ATPs. Okay, Krebs cycle will give rise to 24 ATPs. Okay, overall, and then the energy produced in prokaryotes is 38, whereas in eukaryotes is 36. Okay, so why the two electrons are less in eukaryotes is because for movement of NADH and FADH2, we utilize ATP energy. Where two molecules of ATP is utilized there, whereas prokaryotes don't do that. They're very smart. Okay. So, anyways, anaerobic reactions. Okay, anaerobic reactions, and we have one more part called as what is the overall aspect of this glycolysis? Okay. Is it always the same amount of ATP that is obtained, or is it different? Okay, so it's okay. In the next class when we come, we will take only these 3 slides and we'll discuss this and we'll finish it off. Understood? Fine. You have like 2-3 minutes. You can use it for doubt session. You can ask me if you have any doubts, people. Okay. So anyways, we'll solve these questions in the next class. Don't worry. If you have any doubts or if you have understood, you can let me know. Okay, if you have any concept that has to be revised or something like that, you just let me know again so that we can carry it to the next class and we can do a quick revision.
Sir, you may repeat it complex 1.
Come again?
You may repeat it complex 1.
Hold on. You are asking about some complex. What complex?
Complex 1.
Yeah, complex 1. I should tell about complex 1.
Yes, sir.
Yeah. See, these complexes, what we call it as electron transport complexes. Okay. So we are divided into several complexes. Why we have divided into several complexes? Because see what is happening importantly, the NADH2 that is coming from your Krebs cycle has to be reduced to NAD+. Okay, oxidized to NAD+. Why do we do this? The reason why do we do this is because we want hydrogen atoms. Why do we need hydrogen atoms? When hydrogen atom is moved, see, you can see here very clearly. Hold on, I will just move this toolbar. Yeah, when we see this movement of hydrogen atoms, you can see here from the matrix. Okay, it is moved to the inner membrane. Okay, inner membrane space. As it is moved across, okay, these hydrogen atoms. Now that will result in, see, when we oxidize this, that will result in 2 electrons. So the electrons that are produced have to be moved to the other complexes. Why the electron transport chain takes place? To create conformational changes in your ATP synthetase. Understood? So that is why to move this electron, some gradient has to be produced. No, a proton gradient is produced by movement of electrons. So that is what is complex 1. Why do we have the importance for complex 1? Complex 1 has an enzyme called as NADH dehydrogenase, which will break down NADH to NAD+. Understood? So we will get important factor, that is 4 molecules of, see here, 4 atoms of protons or hydrogen is obtained from this. So now the 2 electrons that are resultant of breaking down this will be used for the transfer of this electron by these complexes. What are these complexes? FES complex and FMN complex. What is FES and FMN? They are called as electron acceptors. Okay, they accept the electrons and they help in transfer of electrons from one complex to other complex. Is it understood? Did you understand?
Sir.
Clear. Okay, fine. Okay, students. Thank you very much. Here we can conclude the session and we can carry out in the next class. Okay. So even it's okay. In the next class, if you have any doubts, you can just put a message and we will solve your doubts. Okay. A very happy new year and have a nice day. Bye-bye.